Have you ever felt it? That sudden, crushing weight in your chest for no apparent reason. That fight or flight response kicking in while you're just standing in line at a grocery store. For decades, doctors have given us the same tired explanation. Your brain is just wired differently, or your amygdala is overactive. But what if I told you that science has been looking at the wrong map? A groundbreaking new study from the University of California, Davis, just published in the American Journal of Psychiatry, has revealed that anxiety isn't about how big or active your brain's fear center is. It's about a tiny, secret group of gatekeepers that have been hiding in plain sight. Today, we're diving into the genetic blueprint of fear and how a single gene called. To understand this breakthrough, we first have to understand the amygdala. Shaped like an almond and tucked deep within the temporal lobe, it's long been labeled the fear center of the brain. For years, researchers believed that if you suffered from anxiety or PTSD, your amygdala was simply too large or firing too many signals. But Professor Drew Fox, the lead author of this new study, says that's like trying to understand why a city has a high crime rate just by looking at the city's total square mileage. It tells you nothing about what's actually happening on the streets. In fact, recent data shows that many people with massive, hyperactive amygdalas live perfectly calm lives, while others with normal structures are paralyzed by fear. This led the UC Davis team to a radical conclusion. The size of the building doesn't matter. It's the people inside the building that run the show. How do you study individual cells in a living brain? You use a revolutionary technology called single-cell RNA sequencing. Think of the old way of studying the brain like a fruit smoothie. Scientists would take a piece of the amygdala, blend it up, and test the liquid. They could tell there was strawberry and banana in there, but they couldn't tell you how many of each or where they were located. The UC Davis team did the impossible. They separated every single cell. They mapped the genetic language of each individual neuron in both humans and rhesus macaques, blind monkeys, because the biology of fear is millions of years old. By comparing the two, they found the core components of anxiety that have survived through evolution. And what they found was a specific cluster of cells that looked and acted like nothing else in the brain. At the very edges of the amygdala, the researchers identified a group called intercalated cells. But in the lab, they have a much cooler nickname, the gatekeepers. These cells are unique because they express a gene called FOXP2. Now, if that name sounds familiar, it's because FOXP2 is famous for its role in language development. But here, at the border of the fear center, these cells have a different job. They act as a choke point for emotional signals. In studies with rodents, these FOXP2 gatekeepers were shown to decide which signals get to enter the amygdala and trigger a panic response, and which ones are ignored. If these gatekeepers are weak or asleep at the post, your brain lets every tiny worry pass through the gate, leading to a state of constant, debilitating anxiety. The UC Davis study proved for the first time that humans have these exact same gatekeepers, positioned in the exact same way. So, why does this matter to you? Because currently, treating anxiety is like using a shotgun to kill a fly. Drugs like benzodiazepines or standard antidepressants affect the entire brain. They make you sleepy, they fog your memory, and they come with a laundry list of side effects because they hit every cell, not just the broken ones. But the UC Davis team found a secret handle on these gatekeeper cells. They discovered that FOXP2 cells are packed with a specific receptor called NPFFR2. This is the moment. By identifying this specific receptor, we can now develop smart drugs, medications that act like a sniper rifle. Instead of numbing your whole brain, these drugs would target only the gatekeepers, waking them up and giving them the strength to close the door on irrational fear. We are moving away from managing symptoms and moving toward fixing the biological gate. Anxiety is one of the most complicated disorders a human can face. It's personal, it's painful, and for many, it feels like a life sentence. But as we map the brain at the cellular level, we are realizing that our emotions aren't just feelings, they are governed by precise biological machinery. The work done at UC Davis is a massive leap forward. It tells us that we aren't broken, we just have gatekeepers that need a better set of tools. If you want to dive deeper into the actual data, I've linked the full study from the American Journal of Psychiatry in the description below. What do you think? Does knowing that your anxiety has a specific cellular choke point make it easier to deal with? Let me know in the comments. And if you want to stay on the cutting edge of how your brain actually works, hit that subscribe button. The future of your mental health might be closer than you think.